My name is Phurpa Wangchuk. I have been working at JSU with Professor Alex Laukas, who is one of the pioneers in hookworm research. Whereas for this funding, we thought maybe we should do something different, focusing on far north Queensland wet tropical plants. So we have been doing some research on those plant extracts from wet tropics, at the same time insect fungi. And we managed to get at least 2 compounds that showed really potent antimicrobial activities against gram-positive and gram-negative bacteria. So this project is it builds upon the funding that Dr. Visai, who is medical student, 4th year I think, who got his funding about 5 grand. So I did all the extractions and isolations, gave the compounds to him, did all the screening. So we finally got at least 2 promising candidates. So we thought maybe we should further this research, try to get some antibiotic drug-lead compounds, so it can be developed into good drug candidates. So that's how this idea came through. From the preliminary data, we found that it is broad antimicrobial. It can treat MRSA, which is good thing. So we found really good activity against MRSA, which is big thing in FNQ, primarily because we live in the tropics. We are prone to that microbial infections compared to other parts of the world. So we thought that is exciting, and also we thought maybe we should, that's what we found, but we wanted to also, based on this, if we get this funding, we thought maybe we should do a little bit extra, focusing on biliary ulcer. We thought maybe we can develop, if we can develop some sort of topical agents, see whether it can kill those things, because we don't have treatment for that. I've been collaborating with some of my colleagues, and they have done some antimicrobial testing in the crude extract, not on the compounds that we have proposed, and they found it has some antimicrobial activity. So that also provided us lead to follow that that basis, because it provided basis for screening. I'm grateful for this funding really, because it laid some foundation. Although it may not be sufficient, we proposed for 25 grants, but at least we can achieve one objective, which is isolation of compounds. More, because what happened is I isolated weak isolated compounds. That was enough to do those preliminary screening. If you isolate compounds from natural products, sometimes you get just minimal quantities, not even sufficient to carry on any experiment. So at least from this base, with this grant, we hope to achieve objective 1, which is to get large quantities of compounds isolated from those extract. Again, we know the plants, we have identified the plants, and maybe a little bit of objective 2 as well, which is to screen for toxicity. Now toxicity is a great thing. If it's highly toxic, we don't want to proceed further, because then it does not serve the purpose. So at least those 2 objectives. So I'm grateful for this fund, because then we are building some foundation, so we can apply for other funding, or maybe same funding next year, and then see where it progresses. If you're talking about benefit not only to FNQ communities but globally, that's how I see. Now what happened is, starting from 1981 to 2014, they have developed a drug lead, novel drug lead or drug entities. So they have developed 1,182 or some, around that figure, and out of that about 73% were based on natural products. It comes from medicine plants, from different insects and insect fungi, all sort of natural sources. Few percentage are based on synthetically produced by pharma companies. So out of 70%, approximately 30% were antimicrobials from natural products. So we are hopeful that we'll get, if we get something highly potent against, especially against MRSA, that can be a big thing, not only for FNQ but also globally, because there's huge, not only demand, but there's huge problem worldwide because of this antimicrobial resistance. It's costing lots for the hospitals to manage that, because you treat once they have to come back again and again. It's hard to treat most of the time. And worst thing is, if you see antimicrobial resistance to the existing drugs, it will be chaotic. It would create chaos not only to FNQ or hospitals or communities, but worldwide. It can damage, or it can be it can halt all the surgeries. You cannot do any cancer treatment as well, because infection is the worst thing you can have if when you have to undergo surgeries. If you have infection that cannot be treated by current antimicrobials, it's a bad thing. So we thought if we can develop something new based on plants, which is already consumed by the people, especially the fruits. So we thought maybe, I think that's a good idea to focus on, carry on this research, see whether we can get anything really good one or not. Yeah. Good thing about these two compounds is that we found is broad antimicrobial, which means at the same time there's pros and cons to it. Broad spectrum means it can treat a lot, but at the same time, if one develops resistance, that can also lead to drug resistance to other strains as well. So it has both pros and cons. Yeah. But good thing is broad antimicrobial. We are hoping that can be good for treating not only MRSA but also other both gram-negative and gram-positive bacteria. This is my first application. I got it, and I'm very grateful to it. That it means a lot. As I said earlier, it builds foundation for future research. So what happened is, if we get enough material from plants or compounds from plants, then and also if we get some good in vitro data, we are hopeful that we can apply for bigger funding that can potentially give us a lot of money to do animal research. And although it may take long, but I think it's worth pursuing it if we find really good activity, without cytotoxicity.